Good day, everyone, and welcome to another screencast here in microbiology. I am your lecturer for today, Dr. Supache Basit, and for today, we will be discussing the microbial virulence factors. And basically, we will answer the question what makes them or these microorganisms more harmful? So, there are several virulence factors that makes microorganisms harmful. One of them is their ability to resist phagocytosis. We all know for a fact that phagocytosis is one of the natural defense mechanisms of our body. Okay, so there are natural phagocytes in our body. So these are the neutrophils, the macrophages, and some of them, such as the macrophages, may even work as an antigen-presenting cell. So these phagocytes play major role in clearing bacterial infections. However, some microorganisms may have the so-called capsule. So capsule, or in some cases, these are not neatly organized. We call them slime layer. So this capsule or slime layer would have the ability to inhibit engulfment. So they will be able to mask cell structures that are recognized by receptors on the phagocyte cell surface. So the one that you can see here is an example of a capsular stain. So a capsular stain, we're able to demonstrate a capsule of the bacteria. So under the microscope, they appear like colorless halo. So what are the examples of bacteria with capsule? So these are Streptococcus pneumoniae, Klebsiella pneumoniae, Haemophilus influenzae. So these are examples of bacteria with capsule. Okay, so there are also some factors that would enable them to resist phagocytosis. So one of them is protein A. So protein A is actually one of the major virulence factors of Staphylococcus aureus. So they are found in the cell wall of Staphylococcus aureus. As you can see here in the illustration, there are two possible functions of protein A. First, protein A will interfere with the binding of the host antibodies to the surface of the organisms. Okay, so it will inhibit antibody response. Okay, so another one is that it will bind also with the Fc portion of immunoglobulin. So this is the immunoglobulin, and then this is the Fc portion, and then protein A will bind here. So it will prevent opsonization, which is an antibody that is responsible to mediate phagocytosis. So protein A will prevent opsonization and eventually phagocytosis by turning the antibody is turned around on the surface. So that way phagocytosis will not be attained by our immune system. So another organ or another part of another virulence factor that may be produced by this bacteria, we call it the leucocidin. So leucocidin is also being produced by the genus Staphylococcus. So leucocidin is otherwise known as the Panton-Valentine leucocidin or the Panton-Valentine factor. Other texts may call it that. Okay. So this Panton-Valentine leucocidin can have the ability or may have the ability to destroy leukocytes. So since leukocytes are destroyed, so it will eventually contribute to the invasiveness of the organisms. So the there are also some factors that can inhibit chemotaxis. Chemotaxis is the ability of these leukocytes to be attracted to substances being produced by bacteria such as the toll-like receptor. So if the chemotaxis is inhibited, the host is less able to direct the neutrophils such as the PMMs and macrophages to the site of the infection. So I have here the list of some of the bacteria and their corresponding ability to interfere with the phagocytic activity of the cells of the immune system. So as you can see, not only Staphylococcus would have that ability, So we have Streptococcus, Bacillus anthracis, Haemophilus influenzae, Streptococcus pneumoniae, Klebsiella pneumoniae, Pseudomonas aeruginosa, Escherichia coli, Salmonella typhi, Cryptococcus neoformans, Treponema pallidum, Yersinia pestis, Mycobacterium, Brucella abortus, and even Toxoplasma gondii. So kindly study this table so that you'll be able to know what are the different mechanisms that would allow these microorganisms to prevent phagocytic activities of the cells of our immune systems, particularly that of the neutrophils and the macrophages. Okay, so there are also structures found on the surface of the bacteria that promote adhesion to host cells and tissues. What does it mean? Some microorganisms would have structure that would allow them to anchor on our cells so that they will not be easily eliminated. So for example, once you're infected with Neisseria gonorrhoeae, these organisms would have Fimbriae, and this Fimbriae will allow the organisms to attach to the epithelial lining of the urethra. So even if you urinate, the pressure of that voiding of urine will not allow them to be easily detached from our tissue. So there are several factors that may be responsible for that, and one of them is called adhesins. Okay, so adhesins are the microbial surface structures that can mediate attachment. So, and the reason why bacteria will actually exhaust the potentials of using adhesins is because our cells, okay, some of our cells have receptor site for the adhesins. So, in some cases, mutation in host or even in infectious agents may result in surface structure change. So, it can be beneficial in some way for us because in this case, adhesion does not occur, or the virulence of infectious agent is affected. So sometimes we may want mutation in the host to occur so that this bacteria will not be able to utilize adhesin as our receptors may somehow have changed physically. Okay, so there has been conformational change in our receptor site for adhesin. So that way, the virulence of that microorganism is eventually stopped. Okay, so aside from adhesin, we also have this Fimbriae. Actually, Fimbriae is actually different from pili because pili are much stronger. So as you can see here in the illustration, that is an example of an electron micrograph view of a bacterial cell with Fimbriae. So these Fimbriae are actually characterized by short, hair-like bristles that surround the bacterial cell surface. Okay, so chemically speaking, it is made up of surface polysaccharide. So again, I actually used this example a while ago. Organisms such as Neisseria gonorrhoeae has Fimbriae. So Fimbriae will enable the bacteria to be attached rigidly to host cell surface structure. So that way, it will increase its ability to colonize, such as in cases of Neisseria gonorrhoeae, causing gonorrhea, and it will provide resistance to phagocytosis. So here is a comparison of a gram-negative bacterial cell wall to that of a gram-positive bacterial cell wall. So as you can see, for gram-negative bacteria, they have a thinner peptidoglycan. So they have non-pili adhesins, they have pilus, okay, and then they have the cell surface polysaccharides, the Fimbriae, and this is eventually not found among gram-positive bacteria, but they have Fibrillia, an example of which is M protein, which is found in some species of Streptococcus. Okay, so some of these organisms would have the ability to survive intracellularly and eventually proliferate so that they would become more virulent. Okay, so there are different mechanisms to prevent organisms from being killed intracellularly because once they are inside our body, it's a total war zone because the cells of our immune system will not easily allow them to proliferate. But some of them will produce IgA protease. So IgA protease is an enzyme that will literally cut the proteins of our IgA. So that will render our IgA useless, and IgA is important for mucosal immunity. Okay, they are found mostly in our secretions. Okay, antigenic variation is one of them, so that they can easily evade the wrath of our immune system. And then some of them would be producing lactoferrin, which can bind free iron, and meningococci or Neisseria meningitidis can actually use lactoferrin for iron. And some of them would have these lysosomes, which will prevent fusion, which eventually escape the phagosome inside these phagocytes, such as macrophages or neutrophils. Okay, so some of them, once they have invaded already our body, some of them would have the ability to penetrate and grow in the tissue by means of the localized infection. So they may colonize few layers or in one area of the body, and then they may cause disseminated. This one is much difficult to manage, because when we say disseminated, it means that it has already spread to distant areas and organs. Okay, so some of them would have the ability to produce toxins and enzymes. Toxins are poisonous substances secreted by organisms. So it could be an exotoxin, or the other one is endotoxin. So let's talk about an exotoxin. So exotoxin is actually secreted by the organisms. And the reason why we call it exotoxins, because these are being secreted into extracellular environment, and they are being released on the upon lysis of organisms. So there's actually a requirement that these organisms would need to be lysed. So there are actually two parts of exotoxins. We have the binding subunit. This will allow toxin to enter our cell, and then the toxic subunits, which will actually disrupt or destroy the cellular. Okay, so what are examples of exotoxins? So most of these are gram-positive bacteria, although there are some gram-negative bacteria here, may have exotoxins. So, for example, anthrax from Bacillus anthracis, Bordetella pertussis will cause whooping cough. So the Clostridium botulinum toxin is one of the most potent in the world. So it's an example of neurotoxin. Clostridium difficile, Clostridium noivii, which can cause gas gangrene, Clostridium perfringens, which will cause gas gangrene, Clostridium septicum, Clostridium sordeli, which can cause gas gangrene, Clostridium tetani can cause tetanus, and Clostridium diphtheriae, Corynebacterium diphtheriae may cause diphtheria. You know, in immunology, you will learn that there is what you call attenuated form of toxin. We call it toxoid, and these are actually being given so that neonates will somehow you'll prevent this neonate from getting this toxigenic bacteria, such as an example of toxoid is DPT, diphtheria pertussis tetanus toxoid. Okay. And then Escherichia coli may cause diarrhea, Pseudomonas aeruginosa, Staphylococcus aureus, Streptococcus pyogenes. All of them may cause pyogenic infection. Vibrio cholerae, of course, the cholera exotoxin will cause a violent diarrhea. And Salmonella typhimurium may cause enteritis, some species of Shigella, and even Yersinia pestis. Okay, so these are the different types of exotoxins being produced by pathogenic bacteria. So screenshot this one. Okay, and this one, on the other hand, is an example of endotoxin. So as you can see here, these are the molecular structure of the endotoxin. So endotoxin is actually composed of lipopolysaccharide. So lipopolysaccharide of the LPS is found at the outer portion of the cell wall of gram-negative bacteria. Because unlike gram-positive bacteria, so let's take a look. Okay, so gram-positive bacteria has a single cell wall. Here, for gram-negative bacteria, you have the inner membrane, and then you have the outer membrane. So the outer membrane for gram-negative bacteria would have the LPS or lipopolysaccharide. So that is the reason why we consider lipopolysaccharide as the major virulence factor of this gram-negative bacteria, because the lipopolysaccharide is the source of the endotoxin. Okay, so moving on, as you can see here, there are actually three regions of the endotoxins. We have here the O-specific oligosaccharide, and then we have the 4-polysaccharide, and then we have the inner lipid A. So the inner lipid A is actually called the endotoxin. Okay, so the endotoxin or the lipid A will stimulate the release of pro-inflammatory cytokines that aid in mounting an immune response. So it's like triggering our immune system to produce cytokines. So whenever these cytokines are being produced, this will of course cause significant dramatic changes in our body, such as changes in our blood pressure, clotting, body temperature, circulating blood cells, metabolism, humoral, and even our cellular immune response, and resistance to certain infection. Okay, so this lipid A will actually trigger our immune system to produce the so-called cytokines. So by the way, in immunology and serology, we will have a separate discussion on cytokines. Okay, so endotoxin will, upon exposure to endotoxin, this will stimulate our body to produce cytokines, and cytokines will actually have hypothalamus as the target organ, and it will raise up our body temperature, thereby causing us to have high fever, and then there will be a drop in blood pressure, so that there's hypotension, and eventually it will result into septic or endotoxic shock, and then coagulation initiation would also be stimulated. This may be fatal, particularly if the DICs or the disseminated intravascular coagulation happens in major blood vessels. Then there would also be severe neutropenia, wherein the neutrophil level would be significantly decreased. In other words, there is what you call immune system disturbance. Okay? So if you will be comparing, therefore, the difference between exotoxin and endotoxin. So for endotoxin, it is exclusively found in gram-negative bacteria, whereas exotoxin can be found in both gram-positive, for example, Clostridium tetani, and even gram-negative bacteria, such as Vibrio cholerae. And then the chemical nature, exotoxin is a simple protein, whereas endotoxin is a complex of protein, lipid, and polysaccharide. And as in terms of stability to 100 degrees centigrade, exotoxin is heat labile, endotoxin is heat stable. We can easily detoxify exotoxin upon treatment with formaldehyde, but not with endotoxin. And then the exotoxin is completely neutralized by homologous antibody, but not with endotoxin. And the biologic activity of exotoxin may depend on individual toxins, because as you can see in the table a while ago, there are several examples of exotoxin. We may have the potent exotoxins, such as the Clostridium botulinum, which is a neurotoxin, and then we have the Vibrio cholerae exotoxin, which will the target organ is the gastrointestinal tract. So the biologic activity depends on what individual toxin are we pertaining to. And like for endotoxin, the biologic activity is same for all types of toxins. Okay, so there you have it. That's the end of our lecture on the major virulence factors of the microorganisms. Be sure that you will be watching the next screencast, which will talk about how would our body defend itself against these invading microorganisms, of course, through the help of our immune system. So once again, this has been Dr. Supache Basit saying, wherever you are, always stay safe and God bless everyone. Bye.